Welcome, everyone. This is Ann Priest. I'm a registered dietitian and manager medical scientific liaison at Nestle Health Science, and I'll be moderating today's presentation entitled Maximizing Health Through the Microbiome, a Life Continuum. Financial support for this presentation was provided by Nestle Health Science. The views expressed are those of the presenter and do not necessarily represent Nestle's views. The material is accurate as the date it was presented and is for educational purposes only and is not intended as a substitute for medical advice. I am very pleased to present today's speaker, Dr. Kelly Tappenden. Dr. Tappenden is professor and head of the Department of Kinesiology and Nutrition at the University of Illinois at Chicago. Dr. Tappenden's research program focuses on intestinal failure, mechanisms regulating epithelial function, and patient malnutrition. For these contributions, she has received multiple awards, published over 100 peer-reviewed papers, and delivered over 500 invited lectures. Dr. Tappenden served as the 33rd president of the American Society for Parenteral and Enteral Nutrition in 2008 to 2009. Chair of the Nutrition and Metabolism and Obesity section of the American Gastroenterology Association Institute from 2009 until 2013, and presently represents the American Society for Nutrition on the Federation of American Societies for Experimental Biology Board of Directors. Dr. Tappenden has been the editor-in-chief of the Journal of Parenteral and Nutrition since 2010. And here are the speaker's disclosures. The objectives of this program today include examining the current impact of the microbiome on health from infancy into adulthood, evaluating various positive outcomes that have been demonstrated when we improve the microbiome, and finally, applying nutrition interventions that have been shown to be positive influencers on the microbiome. And now I would like to turn it over to Dr. Tappenden. Welcome.
Thank you, and thanks to Nestle for sponsoring us and each of you for joining us today. Let's take a moment to orient ourselves to the gut. We know that when it comes to the lay public, they will see the gut as gross, and it is structurally unusual. We have the esophagus as this straight tube coming from the mouth, entering this lopsided crescent-shaped stomach. The small intestine meanders back and forth from the left to the right, looking like beads on a string of pearls. Then it's framed by the colon. But you know what? This structure is exquisitely organized to meet the functional needs of us as individuals. We have that single layer of epithelium, if you zone your thoughts now into the crypt-villus axis, where cells are proliferating from the stem cells in the crypts being engendered to enterocytes, goblet cells, Paneth cells. We have 70 to 80% of the immune cells located there. So it really is precisely organized, despite what one might think with it not being. And what is embedded in this organization is actually this dynamic ecosystem of trillions of essential microbes. It differs along the gut from the mouth to the anus, from the lumen out to the epithelium, and from individual to individual. The number of cells, of microbial cells, outnumber human cells by tenfold. And actually, if we look at the genes contained in the microbes, there's 150 times more microbial genes than there are, in fact, human genes. So when you think of it this way, we're actually a super organism carrying around that incredible microbiota within us. Now, it's really important, as we're going to be able to discuss in a moment. But on the next slide, let's just take a moment to talk about some of the terminology that you might hear. We used to hear microflora, right? Micro being small, flora actually refers to plants. So that was something, if you look at the literature, you aren't going to see much anymore. That's really a term that's been outdated. Biota is what it was originally replaced with, meaning small life, small microorganisms. And now you see biome. What's the difference between the microbiota and the microbiome? The biota is a community of microorganisms. So you can think of those microorganisms much like we would a classroom of students or individuals with a variety on various issues, phenotypes. The microbiome is that community's collective genome. So we see microbiome probably more when we're looking at methods of certain papers. In the research literature, we'll see microbiome because a lot of the techniques we use now are molecular based, and we identify who's there based on their genome. However, when we're thinking about the actual microorganisms and the metabolites that they produce, in that regard, we can refer to the microbiota. Okay, that's the microbes that are present. So both are correct. You would just use one or the other depending on what you're referring to. If it was analyses looking at the genetics, you would refer to the microbiome. Some people think that microbiota is no longer accurate, but it definitely is. Now, because of the methods, NIH launched a 10-year program called the NIH Human Microbiome Project. Now, this was an interesting project that had two phases. The first, Human Microbiome Project 1, was to characterize community groups in the nose, oral cavities, gut, skin, vaginal communities in a healthy cohort of individuals. So they looked at the composition, as I said, microbial pathways, the genomic sequence, so the microbiome, and then isolated various genomes. So this was more to characterize who was there and correlate that with phenotypes. The second part of the project looked at 3 different longitudinal cohort studies. So pregnant women with preterm birth, they looked at vaginal communities there, IBD, looking at the gut communities, and prediabetes, looking at the gut and nasal communities. And what they did was then look at those same 4 things that they did before, but now some of the further analyses incorporating omic analyses. So you can see at the bottom, metatranscriptomics, metaproteomics, and metabolomics, really important features that helps, I think, shift the focus from individual species. Like at first we were hearing more and the analyses were focused on bifidobacteria, lactic acid bacteria, but now there's much more of a recognition that it's the community of microbes that's really important, and in particular the metabolites that they are presenting to that human host. So with that in mind, what you can see is that there are a variety of microbiome habitats. This was developed from the shared repository that came out of the Microbiome Project, which is a central resource for the research community. And basically there's been 4 basic habitats of the GI microbiome. Number 1 here, you would see the upper gastrointestinal tract of healthy individuals, but then that same of individuals infected with H. pylori and often with ulcer disease. Then we have the lower gastrointestinal tract, and for the 4th one is the feces. So this was really important for highlighting the importance of exploring distinct communities. You know, there's this misconception that really the microbiome is within our colon, right? And that somewhere between the mouth and the colon that we get into a sterile environment, or the stomach and the colon, and that's just not true at all. Along the length of the gastrointestinal tract, there is a microbiome, but they're different. And so one of the things we need to recognize when we're looking at the literature is that a lot of it in humans is done on fecal samples. And do fecal samples tell the whole story of everything that's going on in the gut? The answer is absolutely not. But what is it that we know this microbiota is important for when it comes to human health? There are many reasons why the intestinal microbiota is so important. And this was first, if we look on the left-hand side, this information first developed in germ-free animals. So animals that were born and reared in a sterile environment and did not have the opportunity to colonize during that birth process and exposure to the environment. And what was initially noticed is that germ-free animals needed approximately 30% more energy plus supplementation of various B vitamins to grow at the same rate. Okay, so we know that there are certain nutrients that are produced by the microbiota that are important. It also helps regulate energy metabolism. We'll talk a little bit about the obesity literature, but it also extends past the gut into the brain, as you can see, at the central nervous system, and that's really important too. If we move up onto the right-hand side, we know that those germ-free animals are far more susceptible to pathogenic infections than are the conventional animals that have a well-established microbiota. And there's a couple of reasons for that. They both relate to the microbes being present, but then also development of the immune system up on the very top. So let's dig into that a little more closely on the next slide. What we know is that those microbes that are there act as a barrier against pathogens. And that's one of the reasons that a microbiota helps prevent infections with pathogens. So there are intestinal microbiota, as you can see, that are in the lumen, in the mucus layer, attached to the epithelium, and they do all sorts of things that they, as a microbe, are preventing the pathogenic bacteria from being able to come in, take up root. They are competing for the resources, direct competition with those pathogens. They will produce bacterial metabolites. They will prevent the pathogens from adhering and translocating across the microbiota. They create an acidic environment that lowers the opportunity for pathogenic bacteria to infect. And then within the mucus layer, there are antimicrobial substances that are provided. They help maintain that epithelial barrier function, not only by providing the mucus barrier, but also by increasing the epithelial and the tight junctions in between the epithelial cells. So this is called colonization resistance, and it's very important for preventing those pathogenic infections. But it's not just the microbes providing that host defense. On the next slide, what you can see is the development of the immune system is something that the intestinal microbes do. This also helps the host defense because it helps stimulate and mature that gastrointestinal immune system from the time of birth, so in utero, and then out as a child is maturing and aging. So there's a lot of factors there. The epithelium and the immune system is continually sampling what's going on in the lumen and being able to mature, respond to that environment. And that's real important. It's not just being able to see what's there, and defend against it, but also tolerance as it relates to the ability of the epithelium to distinguish between a healthy microbe that is essential versus one that's pathogen. Nutrients, which is, of course, to be taken up by the cell, versus one that is a toxin. Okay, so that maturation of the immune system is all really important and primed and then matured by the presence of that microbiota. So we call that, on the next slide, oral tolerance. Here you can see a study that was done in 166 infants looking at food sensitization as measured by a skin prick test. And what they found was, on the left-hand side, you can see the microbiota at 3 months. And what is thought to be a beneficial microbiota is one with good richness. So one that has high amounts of the enterobacteria and low amounts of the bifidobacteria is one that is dysbiotic. So that's associated with an infant that will go on to be sensitized later in life by 1 year of age. And you can see that enrichment, if you look at the sensitized versus not sensitized microbes at 1 year of age, that enrichment of probio and enterobacteria compared to bifidobacteria being reduced is still present. But the really interesting thing here is dysbiosis is present at 3 months of age, right? So it precedes then the association with oral food sensitization. In fact, as they looked at these data, each quartile of richness, so beneficial microbes at 3 months of age, was associated with a 55% reduction in food sensitization at 1 year of age. So really critical information showing us that this dysbiosis in infants is preceding then the diagnosis of a food allergy later on, or food sensitization anyways. Now, Hippocrates would tell us that all diseases begin in the gut, right? That was a famous quote by him. And certainly when it comes to this type of issue, if we look at allergies or the gut-brain microbiota axis, this is certainly the truth. And we've learned so much about this in the past few years. On the next slide, what this shows is the importance of the gut microbiota with the gut epithelium as it relates to the central nervous system, and the back and forth, the bidirectional interactions in response to that stimuli of diet, meds, infections, where we have these connections that are mediated by the neuronal, immune, neuroendocrine systems, and then resulting in the brain response as it may relate to stress reactivity, mood, sleep, visceral sensitivity. And it really helps bring into the fact that we're talking about something in the intestinal lumen, or that we think is external, related just to the gut. But it's really a systems biology model where we have the impact of the microbes on that whole organ individual at multi-levels of organ systems. So, as I said, over the last 6 years or so, there's been just a blow up of the understanding of the common factors that impact the microbiota gut-brain activity. You can see here on the left-hand side of this image that there's influence of diet, genetics, mode of delivery, as we know, but then the environment, drugs, exercise. And these then are shown to have impact then and associations with changes in food intake, cognitive behavior, stress, as well as social interactions, anxiety disorders. And so when we then look at the next slide, there's several diagnoses that are associated with this dysbiosis that are manifesting in the gut-brain axis, where we have psychiatric disorders, neurodegenerative diseases. You can see anxiety and stress, IBS, pain disorders. You know, it's really quite a broad effect that wasn't recognized until many years ago at all. And it's not just the microbiota that's doing this. We know that there are many influences that are impacting that microbiota on the next slide. What we want for a microbiota is, one, if you look at the green arrow on the left side, a microbiota that is complex and stable. Okay, if we go along this green line on the graph, what you can see is from birth through the rest of life, we want that complex, stable microbiota with lots of diversity, because that will protect against the pathogens. Remember, colonization resistance, in part by training, stimulating the immune function, and of course, supplying all of these important nutrients, interacting with the nervous system. But if we go along the red line, where there's perturbation of that ideal microbiota, this is what we call dysbiosis. Okay, so there's inflammation, oxidative stress, increases in gram-negative bacteria, infection because that colonization resistance and enhanced immunity is not provided, and the metabolites that are produced are associated more with inflammation. So there we have then these perturbations that are associated with infectious diseases, metabolic and inflammatory disorders, much like the chronic diseases that we know are impacting and killing, unfortunately, Americans. So we need to be thinking of these early events that are happening during the first 1 to 3 years as very important, because you can see whether we look at the green line or the red line, this early period is an opportunity where there is a lot of impact on which way you go. Okay, so that's really important. And the influence of those life events are on the next slide, where you can see that the mode of nutritional provision, whether it be human milk or formula feeding, the mode of delivery, the genetics of that individual, of course, the environment, and the antibiotics that are impacted, as well as then the mom, both during pregnancy as well as after, if she's nursing, has on that colonization period. And so getting that infant off to that red or green start, right? And it's so important if we think of the microbiota as a driver of health, because as we mentioned earlier, it's not just the intestinal function and the immune system as we normally think about it. It also relates then to the metabolism that's seen in the liver, as you can see on the left-hand side, or if we move up to the top, the central nervous system function, as we talked about with the gut-brain axis. Okay, so that diversity that we want to be enhancing over the first 3 years of life is really so important, because on the next slide, you can see that in that early period, both prenatal and infancy, and up to 3 years of life, that neural processes occurring are very critical starting out at that point. And then there is programming that is done at that point. And so we want to be able to optimize that there. Not only is that processing time for the brain and neural activity so important, but it's also really important then for that community, or the community fingerprint of the microbiota, because once that's established, it's also relatively difficult to change that. We all have our own community, as I mentioned earlier, and we tend to go back to that. So it takes quite a bit of effort to change that community, even once one has had antibiotics or something or a disease that will knock that out and result in perturbations there. Over time, we tend to go back to that initial community that was established. So keep that in mind as we look at other times of life where it's really important, and we're going to be looking at diseases. So on the next slide, what you can see is a summary of dysbiosis. So what is dysbiosis? Well, if we think of dys, that would be abnormal or less preferential. So we're thinking of a poor community of microbes within the gut, right? And typically on the left is what we would see as an ideal or a eubiotic community where we have obligate anaerobes, so Bacteroidetes, Firmicutes. And then on the right, this is going to be more of a dysbiotic community where we have these proteobacteria, which are facultative anaerobes. So you can see all of the variety of disease states that are present there. Look at this variety. So from necrotizing enterocolitis in preterm infants out through irritable bowel syndrome, inflammatory disease, alcohol usage, toxoplasma infection, inflammaging. You can see various infections, colorectal cancer, even the type of diet that one consumes, that Western high-fat diet, is impacted. So that expansion of proteobacteria, as it's measured in feces anyways, in identifying what a dysbiotic intestine is really important. And we really need to understand the mechanistic underpinnings for establishing this dysbiosis. Some of the literature thinks that it may be weakening of the immune system early on in life, or at various susceptible periods, whether that be by environment or disease state. But we also know that weakening immune system is dependent on the microbes, right? So it's a real hot topic right now, both from a clinical perspective as well as a mechanistic and pathophysiological perspective. On the next slide, I am just showing you a variety of diagnoses associated with dysbiosis in adults. And although it's a little bit older now, it's 7 years back, if you go to this issue, the May 2014 supplement of Gastroenterology, this is going to give you an excellent basis for the gut microbiome in health and disease. It goes through many of these disease states, as you can see, range from diarrhea through IBD, functional bowel disorders, liver disease, obesity, as we talked about, cancer, C. diff disease. And then, so it's a hot topic there in adults, but also in children. On the next slide, you can see there's a number of pediatric diagnoses associated with dysbiosis that are detected very early in life. So if we consider that, you can see that the compositional changes are not just proteobacteria as we looked at the higher level. There are compositional changes that have been further described, and each of these diseases here that you see on the left, or diagnoses on the left, are associated with specific changes in the microbiota. And I show you these not because I think that there's a lot of clinical applicability to that, but to underscore that there are unique community fingerprints associated with each of these situations, and a lot of understanding that can be held. The other thing is that these changes often precede then the diagnosis, as I mentioned earlier. On the next slide, what you can see is the microbial colonization in premature neonates, and that this colonization actually predicts sepsis in preterm infants. So what you see on the left-hand side, well, first, this is a heat map. So for those of you who aren't used to looking at a clustered heat map, don't worry about it too much. What I want you to do is look for trends, and that's really how we look at this type of image. So clustering, as you see more color, you'll know that there's more expression of that certain organism that you see along the bottom. And then the samples are along the right-hand side where you see 1C, 1B, 4A, 6C. And as an infant became septic, they are shown in red. Orange were infants that had systemic inflammatory response syndrome, and green are those infants that were healthy at the time, actually over the course of the study. So there's 3 different types of samples too. The meconium is shown here by the M bar on the left-hand side. Then those infants that were at risk for sepsis are shown here with the R, and those infants that were healthy are shown with the H. Okay, so if we look at them, what we can see is that there were 3 different kinds and disjointed clusters. Okay, we see EM, which is clusters that were enriched in meconium. We see EH, which are clusters that were enriched in the healthy infants. And we see DH, which were clusters that were depleted in the healthy infants. So if we just hit the next slide again, what you'll see here then is the variation that occurred in these 3 different categories, in that there was enhanced diversity, as you can see in the healthy infant species, as well as the meconium from those infants that did well. But those infants that were at risk of sepsis, if we take all of this data, they did not have that healthy community. They did not have the same type of diversity associated with benefits. Now, not only did they have reduced diversity, those were the infants, of course, that went on to develop sepsis, and they were also, those infants that developed sepsis also had more antibiotic use, which could contribute to why there was reduced variation too. So it's very interesting, and it shows us that we need to understand attributes and factors that are contributing to the eubiosis. Like, how do we get infants' microbiome on that healthy green track as opposed to the red? And if we can do so, will we reduce risk? That's really, really attractive, because on the next slide, what you can see is if we look at allergies, so, you know, different disease, of course, from neonatal sepsis and risk of necrotizing enterocolitis, what you can see here are several studies showing the development of allergies or atopy at various stages, with the age of diagnosis being anywhere from 12 months up to 6 years, yet the age where dysbiosis was diagnosed was as early as 1 week, actually, in 2 of these studies. Okay, so well preceding any manifestation of the allergic phenotype. So it's pretty exciting, right? Because it shows us that this is predictive. It may not be causal. What we have is a predictive association here. But it also brings up the idea that if we can identify which infants have this dysbiosis present, can we then have the opportunity to modulate or correct this dysbiosis, and in fact then prevent the undesirable later diagnosis? Okay, so that's where nutrition becomes so important. And on the next slide, what you can see are various strategies for altering the microbiome. Here you can see a schematic detailing various strategies. Direct methods include prebiotics, dietary manipulation, microbiome sparing strategies, whereas indirect methods include probiotics, designer probiotics, fecal microbiome transplantation, and then newer topics that we're just seeing exploited are designing synthetic consortia of quorum sensing and metabolic cross-feeding organisms that really lead to this designer microbiome where there's so much interest right now. Let's start amongst these variety of strategies by talking about probiotics. Okay, so probiotics, of course, are defined as living microorganisms that, when are administered in adequate amounts, confer a health benefit on the host. Now, this in the literature must be validated in properly controlled studies, and there's a variety of mechanisms whereby these living microorganisms are thought to be beneficial. Part of it will not surprise you based on knowing what the microbiota does, but it relates to modulation of the immune function, altered metabolites being produced, crosstalk between the resident microbiota and some of that colonization resistance, interfacing with the host either through metabolic products or adhering to the epithelium, improving gut-bag barrier integrity, enzyme formation. And if we dive into these a little bit more closely on the next image, what you can see is that there are, within each one of these categories, there are specific mechanisms where considerable science has been identified. For example, the microbiota and the luminal milieu, so direct antibacterial, antiviral effects, of course, the production of short-chain fatty acids and modification of bile acids, specific impacts on the gut barrier in B, whether that be stimulating mucus production, inhibiting epithelial apoptosis, increasing tight junctions, stimulating IgA secretion. You could see all of that's going to be impacting innate immunity. And then it goes on and on with enhancing the immune response, neuromuscular function, the microbiota CNS interactions through various neural effects and neural transmitters. So we have considerable basic science on the immunity, metabolism, neuroendocrine pathways, supporting the beneficial effects of probiotics. But I think perhaps controversial to some, where I think we have lesser information or poorer understanding is the translation into actionable randomized controlled trials in human subjects due to the heterogeneity of the literature that's present. And you'll understand what I'm saying or why I'm making that statement in a few minutes if it's not clear right now. On the next slide, where we have good clinical evidence supporting probiotic use is in a few diagnoses. Diarrhea for sure, whether it's childhood, adult, antibiotic associated versus pathogen infection, treatment versus prevention. You can see that there's a lot of studies in the literature. And on the right-hand side, I do want you to pay closely here. I told you not when it comes to the community fingerprints, but here when it comes to the organism with probiotic recommendations, this is absolutely important. So here you can see Saccharomyces boulardii, LGG, Strep thermophilus, L. casei. These are all organisms that have been associated with the beneficial treatment of the clinical condition on the left-hand column. Okay, when we make recommendations to patients about a probiotic, we need to go as far as being sure that the probiotic that we recommend has been established to have benefits in the literature. Okay, we can't send someone off to Walgreens or CVS and have them going up and down the aisles, as I know I have, and I'm certain some of you have too, without specific guidelines on what they should be getting. Okay, some of the probiotics they will find there have no evidence in the literature for certain efficacy. So we need to be arming them with specific evidence-based recommendations. Okay, we also have good data on VSL3 in inflammatory bowel disease, pouchitis, both prevention and maintenance of remission. It's been a little more fleeting for Crohn's disease. I'll show you some data for UC though. Where we have the broadest effect, probably by the broadest amount of species, is in the immune area, enhanced immunity. And that speaks to that overall benefit that the microbes have on host defense. And then we also have very good data in atopy with cow's milk allergy, both treatment and prevention with LGG and Bifidobacteria lactis. Okay, so reviewing these data, this is one of the earlier meta-analyses on the next slide, published, as you can see here in the bottom left-hand corner, in 2002. So, you know, this is a classic paper. And at that point, there were already 9 randomized control trials of fairly high evidence showing that Lactobacillus GG improves clinical outcomes in children with acute infectious diarrhea. Many of these studies confirmed that as rotavirus, though not all. They were assumed to be rotavirus. So what we have here are 9 randomized control trials where it in fact showed that there was reduced diarrhea duration by 0.7 days. And if we hit enter again, what you can see then is that not only was duration reduced, in addition, duration was reduced. So for those, there were less kids that were getting it, but those that did, less kids getting it with the probiotic use, those that did had reduced frequency of diarrhea, and it was deemed to be less severe. Okay, so that was really important, particularly given infectious diarrhea is the leading cause of childhood morbidity worldwide and the second leading cause of childhood mortality worldwide. So really important data there. If we move on to more of an adult, or certainly less of an infant type of issue, here we can look at pouchitis on the next slide, which was also shown in 2014, this meta-analysis with 23 randomized control trials containing 1,763 individuals comparing probiotics with a placebo control in individuals with inflammatory bowel disease who had a recent pouch, and they used probiotics versus a placebo, and found that those probiotics, VSL3 in particular, amongst those that were studied, so any probiotic had a risk reduction of 1.51. So you can think of that as the percentage, if you want, a 49% difference or 51% difference in this case, induced remission. But VSL3 in particular had an even clearer effect with a risk reduction of 0.18. And VSL3 also reduced the clinical relapse rate for maintaining remission in those individuals with pouchitis. So we know that pouchitis is such a big problem in patients who have had their colon out for ulcerative colitis, and that it relapses many times. So this was able to maintain the remission in these patients. So really important. It also has extended that VSL3 data to individuals with mild to moderately active ulcerative colitis, shown on the next slide. So this is a prospective randomized control trial with 147 subjects comparing VSL3 to a placebo control. The primary endpoint at 6 weeks was a 50% improvement in the ulcerative colitis disease activity index. And that was obtained in 32% of the subjects consuming VSL3 versus only 10% of the subjects consuming the placebo. So that was a statistical improvement in the number of responders in the VSL3 group. But if we hit enter again, what you can see is that the number of patients with a decrease in that index of at least 3 points was nearly 52% in the VSL3 group versus 18.6% in the placebo group. So, you know, more than about a 2.5-fold increase in the individuals who had a 3-point reduction, indicating improvements in their disease activity index. And if we hit enter again, those patients that attained remission at 12 weeks in the VSL3 group were 43% of that group versus only 16% of individuals in the placebo group. So VSL3, in the literature, has shown to be both safe and effective at achieving a clinical response and maintenance of remission. These type of data have been harder to show, though, in Crohn's disease. That literature is much less clear than either ulcerative colitis at this point and certainly pouchitis. If we go to the next slide, let's think about antibiotic-associated diarrhea. We know that this is such a big problem, right, when it comes to hospitals as well as in the community. And so what this study aimed to do was look at the evidence for probiotic use in the prevention and treatment of antibiotic-associated diarrhea. You can see here this is well studied. There were 82 randomized control trials with nearly 12,000 subjects, and this was published in JAMA back in 2012, so nearly 10 years ago, which in the probiotic literature is quite a long time ago. The probiotics studied were a range, as you can see here, one of the problems in this literature, and the results were that probiotic administration had a reduction in antibiotic-associated diarrhea of a risk reduction of 0.58, and allowed for conclusions that although there was benefit here, the interpretation by these authors, and I think many of us who study in this area, is that it's difficult to translate very clear effects and benefits in studies like this to clinical protocols and exact recommendations. So yes, a variety of probiotic preparation and use in these individuals did show to be beneficial, but what we don't know is exactly what population characteristics we should be looking for, what antibiotic is going to be associated with the greatest response with probiotic use, what probiotic preparation to select, when to use it, what dose to use, how long to use it. All of that is unclear based on the heterogeneity within the literature. So that's certainly a problem when it comes to translating good evidence into specific recommendations. So let me hear what many of you are doing. When is the best time, in your opinion, to use probiotics in patients taking antibiotics? So there are a bunch of answers for you to select here. What I'm going to do is ask Anne to come on because what she can do is let me know what the results are as all of you take the opportunity to respond to when the best time is to use probiotics in patients taking antibiotics to mitigate the effects of the negative effects of antibiotics.
Okay, so we are getting the results, and currently the top answer is 43% have said as soon as the antibiotic starts, and then close behind with 33% prior to starting an antibiotic. And about 20% are saying once antibiotics are finished.
Okay, thanks, Anne. That's really helpful, and I think that there's some rationale for each of those answers. Now, prior to starting antibiotics, we don't have real good data on. But what I can tell you is that on the next slide, what we do have are data from a very nice study, meta-analyses published in Gastroenterology in 2017, looking at prevention of C. diff infection. So, really one of the fears when we're using antibiotics in hospitalized patients. So not just antibiotic-associated diarrhea, but in fact that hospital-acquired infection, which is so difficult and such a negative outcome now with some of the policies that have been put in place with the Affordable Care Act. We really want to be trying to prevent these hospital-associated infections like C. diff. So in preventing C. diff infection in inpatients, probiotics were compared to placebo control. And what you can see here in 19 randomized control trials with high quality of evidence in 6,261 participants, that there is a great reduction in the risk of C. diff infection with probiotics compared to not. So 16 per 1,000 with probiotics compared to 39 cases in 1,000 without probiotics, and that the adverse events were not different between the groups. So there was not a situation where the probiotics were causing a sepsis, as is sometimes a concern that's not established by these literature. The interesting thing is that these authors also did a secondary analysis looking at the timing. And one of the important take-home messages from this study is that if you compare the studies where the probiotic was used on the first or second day of antibiotic use compared to the 4th to 7th day of antibiotic use, that there was a significant reduction in C. diff infection incidence. So that sub-analysis was really important. Now, of note, they also did a sub-analysis on species of probiotic, dose, formulation, duration, all of those issues we talked about being a problem in the literature. This literature were too diverse to draw any conclusion on those things. So the only conclusion by that sub-analysis was using it earlier. As early as the time that antibiotic is started is important. I have people ask me, well, why is a probiotic even effective early on? Because isn't the antibiotic killing it? And I think the answer there, that's a valid concern, and we don't know for sure. Certainly there's designer antibiotics that are being developed to try and protect probiotics at the time. What we need to recognize, though, is that antibiotics are not impacting every single microbe within the gut, but that they're impacting some and reducing that diversity and abundance. And so various probiotics are able to provide some benefit during that time. Okay, so let's move on from one important problem in the hospital to another on the next slide, particularly as we think of the last 2 years and COVID, right? So critically ill individuals. What you can see on this image is that there is a dysbiosis that is occurring with critical illness, and it's actually related to the severity of that critical illness. So the two bars on the left in blue are healthy volunteers, whether they were exposed to the hospital on the right because they work there or not on the left. But then when we look at patients staying in the hospital, so green, those that are ill, not just exposed to the hospital, as are the hospital workers. Those that are minimally ill don't have any effect, but as we move into moderately ill and critically ill, we have a dysbiosis that's developing, particularly in those critically ill individuals. The altered ecosystem that's actually happening very quickly in the course of their disease, some studies showing in a matter of hours, is a difficult situation. And so that really makes us wonder, is there something that can be done here with diet or probiotics? And on the next slide, what you see is a meta-analysis published in March by the Canadian Clinical Practice Guidelines Committee. They had last done a meta-analysis in 2018. Now there are 33 studies, and what they found is that the addition of probiotics to enteral formulas, so to enteral nutrition, had no impact on overall hospital or ICU mortality, had though an impact in reducing infectious complications, which is what you see here in this forest plot. It tended to reduce ventilator-associated pneumonia, though they were underpowered for that, but the relative risk was 0.74. And there was no effect on hospital length of stay, but there was also a numerical reduction in ICU length of stay that was not yet adequately powered. No effect on diarrhea in these ICU patients, which we know is a problem based on tolerance to enteral nutrition or feeding intolerance. But there is a benefit on the ICU populations, and then appears to be ventilator-associated pneumonia in particular, as well as then the ICU length of stay. So those last two require additional data. Okay, so the last disease, as we can classify it now on the next slide, relates to high-fat diet-induced obesity and diabetes. So metabolic syndrome, chronic diseases that are killing Americans. And this is a summary of some mechanistic work done in animals that is supported by what we know in epidemiological data. So there are clinical outcomes that are consistent with this, but these mechanisms are so fascinating that I just wanted us to take a look at it. Now, these are data that were collected in OB mice. So these mice are interesting. If you feed them a low-fat diet, they grow like any other mouse. But if you feed them a high-fat diet, a Western diet, then they become obese. Okay, so it's particularly relevant. Starting from the top half left-hand side and moving down, let's go from panel A through G. What panel A shows us, just that red, those animals fed the high-fat diet versus the OB animals that fed the low-fat diet, or those animals fed a high-fat diet, but the addition of VSL3 did in fact increase body weight. So high-fat diet versus low-fat diet did exactly what we know this model will do. They increased their body weight. They became obese. But if you added VSL3, as shown in the light blue, that mitigated that obesity from developing. Okay, very interesting. On B, what you can see is the fat mass in that, yes, fat mass here with red, the high-fat diet was significantly greater than control, but again, mitigated in the brighter blue, in the bright blue color, when VSL3 was added to that high-fat diet. You can see the adipocytes being significantly greater in circumference with the high-fat diet, but not when VSL3 was added. And if we look at D and E, those panels looking at at a glucose tolerance test and an insulin tolerance test, respectively, show better glucose control and insulin response in the control versus the high-fat diet group, wherein the dysglycemia is corrected when BSL-3 is included with that high-fat diet. Okay, so it's not different from the control and shows much better effects than when the high-fat diet is fed alone without that BSL-3. This extends then not just to systemic glucose control, but also to liver, where in fact you see that that high-fat diet resulted in hepatic steatosis that did not develop when the high-fat diet was also combined with BSL-3. And all of this relates then to cumulative food intake, wherein the BSL-3 supplemented group was not different from the low-fat diet control, but those animals having the BSL-3 or the high-fat diet alone did have increased appetite and food intake. Well, food intake in these mice, it does correlate though with reduced measures of appetite and increased measures of satiety in human controls in human subjects looking at these markers. So, it's really interesting the different types of evidence, solid physiological evidence we have here. And this study, if you're interested in going to it, this group has followed up and has shown that if you hit enter on the right-hand image here, what you can see is that high-fat diet, when combined with BSL-3, is associated with organisms that are producing more of the short-chain fatty acid butyrate, which is stimulating the endocrine L cells in the distal gut to secrete more GLP-1, which you will know of certain pharmacological fame now when it comes to treatments for type 2 diabetes that also are entering their way into weight loss treatments, resulting in a lean phenotype. So really interesting data looking at new advances in clinical protocols for both type 2 diabetes as well as obesity that are related back to basic science supporting dietary fats as well as then impacts on the microbiota. A fascinating area to study, right, or to follow in the literature, even if you aren't looking at it in the lab. So I want to shift our thoughts now from probiotic therapies to prebiotic therapies. Okay, we have a new definition since 2017 on what a prebiotic is. It's important to note that the term prebiotic was not actually coined until 1995. It's a relatively new term compared to probiotic, which was coined 100 years earlier by Eli Michnikov, who was the Nobel Prize winner for a microbiologist who established phagocytosis and went on then to study the impact of various microbes in the human colon at that point. So a prebiotic at this point is defined as a prebiotic is a substrate selectively utilized by host microorganisms, so selectively conferring a health benefit. Okay, so what this is an expansion of previous definitions that allows for inclusion of non-carbohydrate substances now, because we know that prebiotics can extend into fatty acids, for example, specific fatty acids, exert benefits beyond the gastrointestinal tract. It used to be limited to the gut. Now we know it can be other organ systems too, like for example, when Jennifer Aniston tells us about prebiotic in moisturizer, that now is included within a prebiotic type of benefit. It can include other substrates than food. So again, moisturizers, I've seen lipsticks, all these types of things, right? And it can apply to animals and not just humans. So when you see your cat or dog food containing prebiotics, that's why this new definition allows for that too. So it's true that most prebiotics are in fact dietary carbohydrates that are non-fermentable in the gut, but since 2017, that's been expanded and allows the inclusion of many other nutrients as well as applications. So I want to do another poll, and I'm going to call on Anne again. on the next slide, please tell us how do you beneficially impact the microbiota in your patients? Do you use plant food-based diets, encourage consumption of fermented foods, do you use fiber or prebiotic supplements, perhaps medical nutrition foods or enteral formulas, oral nutrition supplements supplemented with probiotics or prebiotics? And maybe you don't target the microbiota at all in your current practice, but give us a sense for what it is that you do currently. And Anne, I'll let you let us know when we have some results.
Okay, we will give it a second. And let's see if we can get our results up here. Okay, so it looks like 38% of the attendees are saying that they would recommend a fiber or a prebiotic supplement. Following that, 23% a medical nutrition food with a probiotic or prebiotic. And then lastly, at 19%, a plant food-based diet.
Okay, very interesting. And I like all of those answers because depending upon our area of practice or the client or patient that we're working with, those can all be very relevant. On the next slide, I just want to spend a couple minutes talking about prebiotics because just like we have to be specific on our selection and advice regarding probiotic use, I believe we have to do the same thing with prebiotics. Okay, and so I'm often asked, well, what is the difference between prebiotics and dietary fiber? And we can come back to that. Let's talk about, well, prebiotics and dietary fiber. In not all fibers have prebiotic activity, okay? So you can't use them interchangeably. Some fibers do have prebiotic activity and they can be called a prebiotic fiber. But also, not all prebiotics are fibers. We know that we have prebiotic action of specific fatty acids, as I mentioned earlier. What about human milk oligosaccharides in human milk? We know that those aren't dietary fibers, right? But they do have prebiotic activity. So the two terms can't be used interchangeably. And when it comes to prebiotics, one kind does not fit all. You can't just throw in a prebiotic without carefully selecting it, the same as I've argued for probiotics. So just to discuss two of the most common kinds here, galacto-oligosaccharides or galactans, fructo-oligosaccharides or fructans, are two types of prebiotics. If we just use FOS as an example, what we have here is a sucrose molecule, so you see glucose and fructose, with various lengths of fructose polymers conjugated onto the end of that sucrose molecule. Okay, now these come from plants. Chicory, artichoke, banana, onion, garlic are all sources of this. And sometimes you'll hear them referred to as inulin type prebiotics. Sometimes you'll hear a short chain prebiotic with just 2 to 5 units of FOS conjugated to that sucrose as short chain FOS. Sometimes FOS alone, sometimes FOS alone. So if you're using a short chain FOS, you should say short chain FOS or FOS. Oligofructose is when there's a medium chain of fructose units, so much like an oligopeptide would be a medium chain of peptide units or of amino acids. And then inulin is long chain, okay, greater than 10 units of that fructose polymer, but can also then be sometimes called long chain FOS. Okay, so you need to be specific in what you're calling it, and why you need to be specific and select specifically is that the way microbes access these molecules is different, even though it's the same chemical structure that they're dealing with. The microbes come in and start fermenting along the fructose end of the polymer. Okay, so they can't access the sucrose end, and there's only one end on these linear prebiotics. Okay, so if you have a bunch of short chains, a gram of short chain fructo-oligosaccharides versus a gram of long chain fructo-oligosaccharides or inulin, or particles in that gram of short chain versus the gram of long chain particles, right? It's like if you put kids in an elevator versus adult in an elevator, you can get many more children in because their weight is so much less than the adults, right? Now, because there are so many more short chain molecules within that equal mass, many more microbes can come and start fermenting at the same time because there's many more chains. You can think of this as like a ticket wicket at Disneyland. If there are more ticket wickets open, we don't have to line up as much, right? But if you're working with the long chain, then there's going to be fewer molecules and the microbes will have to queue up. What does that mean? The short chain is fermented much more quickly and much more proximally in the intestine than is the long chain. It's going to be fermented much more slowly and evenly along the length of the gut because the microbes are having to queue up and they don't consume it nearly as quickly in that fermentation process. So where would you want to use different ones? Well, if you're looking for just a generalized type of gastrointestinal benefit, well, then go for a longer chain or a mixed prebiotic source. But if you're looking at a patient that has proximal type of effect, or you're, for example, a short bowel syndrome patient that has very rapid transit time and we're trying to produce that beneficial nutrient butyrate, we want to do that as quickly as possible because their transit time is so fast, right? So in that instance, you definitely want a short chain fructooligosaccharides because it will be fermented much more quickly and thereby fermented in the patient's intestine. If we use a long chain fructooligosaccharides, that's going to be fermenting in that patient's ostomy bag or, after it's been excreted from that patient's body. So not providing the same benefit at all. So please be specific. One fiber or prebiotic does not fit all. And we want to be thinking about that benefit. Now, in distinguishing between the two, I do want to say that most fibers do provide general stimulation to the microbes within the gut. Why they don't have prebiotic activity necessarily is because, remember, a prebiotic involves selective stimulation of those beneficial microbes. So general stimulation by changing transit time or increasing bulk or any of those other things that fibers typically do doesn't meet the prebiotic criteria. In order to have prebiotic effect, it needs to be that selective stimulation. Okay, on the next slide, I want you to leave today really recognizing that prebiotics may be 100 years younger than probiotics. They have far less studies in the literature, and some of our non-nutrition colleagues will say that they're less exciting, particularly if you call them dietary fibers. However, they are very important. On the next slide, I've summarized some key reasons why I want you to consider prebiotics and the important benefits of those. If we compare the prebiotic literature to the probiotic literature, and I'll show you this in a few minutes, we can evoke very similar benefits with prebiotics as probiotics. Here, what we're providing is a substrate for the microbiota that helps have lasting effects on the microbiota and the associated clinical benefits. When one consumes a probiotic, they will achieve the benefit if it is, in fact, a properly chosen probiotic for a certain diagnosis. They will receive that benefit while they're consuming that probiotic, but there are not lasting impacts on the microbiota. That probiotic will only be shedding, and this is pretty well established in most cases. That probiotic will have its benefit and be shedding from that individual while the probiotic is being consumed. But when one stops consuming that, the probiotic goes away. It typically does not take up residence within that individual's microbiota, and therefore the benefits of that probiotic often go away. I'm going to show you evidence where that is not the case with prebiotics. We can use prebiotics to foster and cultivate the type of microbiota that has lasting outcomes and benefits. And finally, when we talk to our non-nutrition colleagues, and in some cases have reasonable concerns on probiotic use in preterm infants or critically ill patients, what we can have confidence on is that we're not talking about a live bacteria here. We're talking about a safe, evidence and food-based strategy that's incredibly inexpensive and associated with value. So we're talking about a real different proposition here. Now, on the next slide, one of the things I want you to leave with as we think about the specifics of prebiotics is that they are all different too. We talked about it from a fermentation perspective, but here if you look at inulin versus pectin, so inulin, that long-chain fructan versus pectin, we know often coming from citrus fruits, a fiber with prebiotic activity. You can see here that among the top 20 most abundant type of microbes, and here we're talking about operational taxonomic units, OTU. That's not what's important. What I want you to see is that inulin will have various changes on that community profile versus pectin will have different ones. So you can't just use them interchangeably, because what we know is that various diagnoses have fingerprints that we talked about earlier, right, of that community changes. And here, as you can see, various prebiotics will impact those communities differently too. So to move backwards from what we did with probiotics, let me show you some similar data on prebiotics. And I know I only have 10 minutes left, so I'm going to hurry a little bit if you don't mind. On the next slide, what you see is the impact of prebiotics on obesity using many of the same mechanisms here, wherein that high-fat Western diet will lead to the same pathophysiological consequences, but that if we add in a prebiotic, an inulin-type fructan, which is what the ITF is, or the AX, the rabinoxylin, which is another prebiotic, you can see that the metabolic changes at the various targeted organs result in many of the same mechanisms, resulting in a reduced appetite and reduced obese phenotype that's supported then by epidemiological data with a high-fat diet. If we move on to that and look at the other example, which was critically ill patients, here are the summary questions from the Aspen SCCM guidelines for fiber and prebiotic use in critically ill patients. Should we be using fiber additives routinely in hemodynamically stable patients on standard enteral formulas or in diarrhea? And what they suggest is yes, that the expert consensus, so there's not strong literature in this area, but the expert consensus is that a fermentable soluble fiber additive, now they've got their fermentable soluble versus prebiotic a little messed up here. As you can see, they have fructans and inulin as different things. You know better than that now, but that yes, we should be considering routine use in all hemodynamically stable MICU and SICU patients. They suggest 12 to 20 grams of a fermentable soluble fiber supplement be used if there's evidence of diarrhea too. Now, the Canadian practice group, the guidelines were updated here in March 2021, and they do a better job of citing the evidence. Now, they're 5 years down the road, so there is more evidence. There's 12 studies that they reviewed. And interestingly enough, their conclusions are here at the top. No evidence on diarrhea, despite there was a significant reduction in diarrheal days, and that there tended to be a reduction in the patients with diarrhea of 50% that was shown, may be associated with a reduction in mortality, 46%. It was underpowered, but that's a pretty important numerical reduction, right? Hospital length of stay was reduced numerically by 4.1 days, and ICU length of stay by 4.3 days. Now, granted, these did not reach statistical significance, but given that we're only talking about 12 patients or 12 studies and 1 to 200 patients, depending on what outcome you're looking at, I think it's very hard for us to overlook these numerical differences, particularly when we have a DRI for dietary fiber and it has been established as a nutrient that we as humans should view as essential with regard to human health. Why would we not be using it with our critically ill patients, given such data like this, particularly when we know it's underpowered? Certainly we need to be doing more focused studies in this regard, but I don't think we can overlook those data or the DRI. Now, if we move on to the next slide, which looks at enteral formula supplemented with a prebiotic on diarrhea in neurocritical care patients. This was prospective randomized control trial to look at standard formulas versus one supplemented with prebiotic, and what was used here is 3.5 grams per liter of short chain phos for 3 weeks. That prebiotic formula allowed for increased administration of the formula, and we know that getting energy and nutrients into these patients is so important and often difficult given the enteral intolerance in critical care patients. So that there was a higher rate and early achievement of the target dose, statistically so, in the prebiotic group compared to control, and that there was less diarrhea. Look how much less, 9% in the prebiotic group versus 57% in the control group, as well as though that 9% of patients consuming prebiotics, it was faster to resolve than the group consuming the control formula, and many more of them had that consequence. So you can see a benefit here in critically ill, similar with prebiotics. And on the next slide, if we look at antibiotic-associated diarrhea, here specifically those patients with C. difficile diarrhea, we know that relapse is such a problem. 20% of these patients relapse. So in this study, looking at prebiotic oligofructose, you now know that to be a medium chain fructan, that when antibiotics are used, reduce the relapse rate in patients. Those that did relapse had to stay longer than those that didn't, 53%, but that the relapse was far less common, 8.3% in the prebiotic consuming group versus 34% in the placebo formula group. And of course, those that did get diarrhea, if they were consuming the prebiotics, it settled within 3 days versus taking 6 days for the control group consuming, or the control consuming group. So here, very similar story as I showed you with the probiotic literature, right? And where we can see these lasting benefits is on the next slide. If we look at that susceptible time frame of infancy, you can see here that when we feed children a formula, or infants a formula versus human milk, we know that they have an increased incidence of infections, allergic or atopy that develops. And here, looking at a control formula, overall, any kind of infection was reduced in the short chain GOS, long chain phos consuming group compared to placebo. That was statistically different. And the same can be said for upper respiratory tract infections. This study was underpowered for any of these other specific types of infections, but you'll see numerical decreases. And when it came to infections requiring antibiotics, that was significantly lower in children who consumed the short chain or the prebiotic formula, and those prebiotic consuming infants also had fewer episodes of fever recorded by their parents. Now, what's really fascinating about this is I am showing you data here on this table when these children were 2 years old. These data were also very similar after 6 months of life, when all that had happened was the formula versus the placebo. But once those children were 6 months old, they started consuming whatever their parents fed them or they chose to consume. And the benefits here were extended through that first 2 years of life. So again, not what we see with a probiotic, where those benefits in this example would have stopped at 6 months. Here, they're extended out through 2 years of life. And A follow-up on this study also showed that there was less manifestation of the allergic phenotype, in that at 2 years, those infants that had consumed the prebiotic formula during the first 6 months of life had less eczema, less hives, less wheezing. And I'm not going to spend time going through this next study. I'll let you have it as a reference, but it shows the same type of thing in what is called the PIPA study, the prebiotic in the prevention of atopy, wherein there were beneficial changes that were seen early on at 48 weeks into a trial, and then at 24 months, there was still sustained benefit. So well after the formula consumption and treatment had stopped. So this leads to some allergic guidelines or allergy prevention guidelines that were published in 2016 that are quite interesting. So what these McMaster World WAO McMaster University guidelines did was look at various factors impacting the development of allergy in children. Here is the statement for probiotics. Because of the impact of increased propensity for allergies developing in women or parents who have a high risk of a diagnosis of allergies themselves, they recommend, based on all of the literature, that in infant or in pregnant women who are at high risk for having an allergic child, to use a probiotic when they're pregnant, to use a probiotic when they're breastfeeding, and also to use a probiotic in those infants who are at high risk. And they state that these recommendations are conditional and supported by very low quality evidence. Let's contrast that on the next slide, where we have prebiotic evidence that are included. And what they say here, based on much better evidence in prebiotics compared to probiotics, is that they recommend that prebiotic supplementation be used in all infants, not exclusively breastfed. So if an infant is receiving a formula, whether it's partial or sole source, that should be a prebiotic supplemented formula, whether they're at high risk for allergies or not. The prebiotic literature is that clear for a benefit. Here for reference is a similar infectious diarrhea benefit of galacto-oligosaccharides, so that medium chain galactin, reducing the incidence and symptoms of infectious diarrhea. On the next slide, I'm showing you dietary inulin, so the long chain fructan, reduces the inflammation associated with pouchitis. So just very similar to what we reviewed for probiotics, but achieved with a nutrient, right? A safe nutrient. Now, sometimes on the next slide, you will talk to your non-nutrition colleagues and you will suggest a probiotic, and they will look at you and say, no. And they'll cite this ProPatria trial, wherein individuals, patients with severe acute pancreatitis, were in a trial looking at an enteroprobiotic or placebo twice daily for 28 days. And this study was shut down early because the treatment group with the probiotic died earlier. So the treatment was causing increased death or associated with increased death. And one of the things that I want you, besides all sorts of critiques on the ProPatria trial, one of the things I want you to listen for is the fact that you suggested a prebiotic and they responded with concerns about the probiotic. You said PRE, they responded with a PRO. Okay, this is a really common thing that I found with our non-nutrition colleagues. They aren't real familiar oftentimes with a prebiotic. They may not even know what it is, although it's becoming more and more common as part of our even grocery shopping or moisturizer type of exposure, as I mentioned earlier. But what they will hear is, for example, I've given many hour-long lectures on prebiotics, and when the question period comes, I get a bunch of probiotic questions. Now, in this instance, we're talking about both, right? But that even happens when I'm only talking about prebiotics. People hear prebiotic and they think probiotic. So you have to point out to them the difference and the safety and what your recommendations are. Now, what about, just to finish off in our last couple of minutes, and I promise I won't take much time, can we achieve the same thing with a food-based diet? On the next slide here, you see that there are many of the mechanisms that we talked about in the OB high-fat diet fed mice that result in changes in the liver and inflammatory metabolism and increased barrier dysfunction. And that relates then to the gut microbiota. And on the next slide, what you can see is that short-term alterations in diet do impact the gut microbiota. Here you can see the impact. These graphs show fiber intake that increases on 2 weeks of a plant-based diet within 2 weeks, but you can see it even within 1 week. There's great increases here within a few hours on a plant-based diet versus reductions in fiber intake on an animal-based diet. These are associated with changes in a more dysbiotic community in the animal-based diet. And of interest, if we look at fat intake, these would just look the opposite of what we see here on fiber intake. So that animal-based diet is associated with increase in bile-tolerant microorganisms, reduced firmicutes that metabolize dietary plant polysaccharides and are thought to be those beneficial microbes. A link between dietary fat, bile acids, growth of microorganisms capable of triggering inflammation. Okay, so these changes are impacted by diet, and they do so very quickly. These next references, the next slide, go and look at these studies because this really gives us the future. They've looked at a variety, the literature of a variety of different diets that you see along the top. The best impact that's available in the literature is with the Mediterranean diet. Please note this also includes vegetarian diets, low FODMAP diets. The Western ketogenic diet doesn't surprise us at all, right? But that Mediterranean diet is associated with the best impact on microbial diversity and stability associated with health benefits. And the future of our research on the next slide is so interesting. These studies coming from Jeff Gordon's lab, what they've done is they've taken human gut microbes, fed different combinations of 34 different fiber preparations, and then colonize those. So in a notobiotic mouse, so that mouse is then going to harbor that human microbiota that's being fostered with the different 34 combinations of fibers. And then they're looking at the ability of the microbes that are there to process different types of fiber glycans or carbohydrates, what strains and enzymes are activated, so that we can have a personalized approach based on the variety of one's diet into the changes that may be made. And they're using these really cool bead modified nutrients so that they can really use them as biosensors of the carbohydrate metabolism that's happening and show us what microbes are fermenting what fibers. And it's leading to microbiome engineering. On the next slide, you can see is leading to personalized responses that one can try and target based on the community present within that individual as it relates to changes in the dietary timing, whether that be season, the circadian rhythm, things like intermittent fasting or differences in their microbiome, and then trying to understand the different diet that they may have based on that interpersonal variation, so that personalized nutrition can really become a thing that we're going to be moving towards, even on the next slide with microbiota-based diet disease. So we can look at the clinical, microbiome, genetics, personal goals, lifestyles, put that into machine learning with artificial intelligence, and understand then the metabolites, the community of microbes as it relates to that specific individual's community or disease susceptibility. The opportunity here is so consistent with the goal of personalized nutrition, but not limited to disease control and prevention, but perhaps the modulation of physiology to achieve a particular lifestyle like that would be associated with athletes, for example. So it is relevant that we think about prebiotic and fiber intake on the next slide. I've told you how I think we need to be thinking about that when it comes to those patients we're feeding in the hospital, but we also need to be thinking about that when it comes to patients consuming food, whether in health or disease. We know we aren't meeting these guidelines at all, and there should be encouragement for all individuals to be doing that during health and disease. Whether if there's a contraindication, then I think we can look at fiber-free formulas in those we're feeding enterally, but otherwise we shouldn't. To end, we must be thinking about this biodiversity concept, which on the left is much more like a meandering stream rather than one where it's just being plowed through, and we don't have the diversity along the length of the gut in the various environments. Because what's going to happen then is that we aren't going to have the dietary fiber fermentation, the change within the microbiota that's present, and we won't be able to have the benefit from that robust, stable, and complex microbiota. So with that, I want to stop and leave you, I hope, with the concept that the microbiota is associated with so many important physiological benefits in humans, and that nutritional strategies employing probiotics, but also very importantly, prebiotics, and sometimes symbiotics, which we didn't even get to touch today, are important for stimulating that microbiota, fostering the optimal community, which is going to be different amongst each of us, but will provide the critical functions then resulting in enhanced human health and hopefully resistance to disease, or alternatively treatment in the situations where we know that there can be benefit. With that, I'll stop, hand it back to you, Anne.
Well, thank you so much, Dr. Tappenden, for that excellent and timely presentation.